Hello, this is Justin Priest in the JSWell lab at Bean Project, and today I will be introducing the beta release of the planned Reactome web application. We'll be discussing the general nature of pathway databases, the Reactome software platform and data model, our implementation of Reactome, the plant Reactome, using rice as a reference species, and take a look at a few examples of rice pathways. And we'll finish it up with a discussion of future development. Pathway diagram illustrations have long been used by scientists to help visualize abstracted models of closely related biomolecular reactions in organisms. Pathways that have been modeled on computers can make it easier to curate, maintain, annotate, and analyze experimental data, such as gene expression assays. One such computer-enabled model visualizing and analyzing pathways is Reactome, a curated open-source and open-access pathway database that can be found at reactome.org. The initial Reactome project focused on human metabolism, signaling, and gene regulation, and it now has over 1,400 pathways for Homo sapiens. Human is the reference species at reactome.org and has been extended to other species on the basis of gene orthology and inferred reactions, primarily generated through OrthoMCL and Compara.
The Reactome data model is organized around the fundamental building block of the reaction. It depicts biological processes as a set of connected reactions, and therefore it can represent many events and states in those processes. In addition to standard biochemical reactions, changes in state such as binding, translocation, and degradation can also be represented. These individual reactions are then chained together into larger networks to form pathways. Reactions in Reactome are typically depicted in the standard model of an input or inputs encountering a catalyzing agent and generating outputs as a result. There is also an extensive set of diagram elements to represent small molecules, proteins, complexes, protein sets, and cellular locations. Additionally, there are multiple types of reaction node that allow you to capture state, such as transition, binding, and dissociation.
Now we'll turn our attention to the Gramine project's implementation of the Reactome platform, the plant Reactome. Our pathway data for this initial release represents Rhiza sativa. Our sources for this data come from the Gramine's version of RicePsych, release 3.1, and also orthologically based pathway projections from Homo sapiens in the human Reactome project. Our initial rice pathway data is organized into two main groups: rice cellular processes, or regulatory pathways, and rice metabolism, containing metabolic pathways. Within these two main groups, pathways are organized hierarchically into superpathway families, such as translation, and in this example are further subdivided into the initiation, elongation, and termination steps. Further pathway subdivision is also possible. Looking at a specific pathway in the translation initiation process, the formation of a pool of free 40S subunits, we can see that it contains multiple reactions and various constituent complexes and a protein. COP2-mediated visible transport is an example of a more complex pathway involving multiple cellular locations, reaction types, and participants. If you select a specific reaction on a pathway, such as this phenylpropanoid biosynthesis pathway diagram, you have access to details about that reaction, the inputs, outputs, catalyzers, molecular functions, and the organism that the pathway belongs to, along with a list of participating molecules. Now that you've had an introduction to how pathways and reactions are organized in the plant Reactome, I'll speak briefly about the nature of the data conversion process we followed to get rice pathway data into this environment. The bulk of our pathway data came from RicePsych version 3.1. We exported this data into the BioPax format and ran a data conversion utility written in Java to convert that BioPax data into the Reactome data format. We also enhanced this data by updating Kebi references to reference molecules and undertook a review of the UniProt assignments for the Japonica proteome. Finally, we laid out the pathway illustrations using the Reactome Curator tool desktop application and imported all of this resulting data into a plant Reactome database. Once we had all of this converted rice pathway data, we placed it in a new plant Reactome database and installed a new instance of the Reactome software platform and began the process of customizing that to function for plants specifically. Let's take a look at some examples of metabolic pathways in the plant Reactome as we proceed with a tour of the plant Reactome beta site. When you visit the plant Reactome beta site, currently located at plantreactome.oicr.on.ca, you'll see the plant Reactome front page with a standard menu at the top, a pathway search box on the left-hand side, a featured pathway of interest on the right-hand side, some general project information and recent news and notes center at the bottom, and particular pathways of interest on the lower left-hand side. The pathway search box allows you to search for particular pathways, reactions, and genes by name, and will take you to detailed search result listings that would allow you to further click through to the actual pathway diagram on which that gene, reaction, or the entire pathway itself is found. You can also get generalized statistics on the basis of more general searches such as glucose that will show you all of the various matches across the entire plant Reactome database. There is also an advanced search feature found under Tools in the main menu that will allow you to enter multiple field constraints at the same time, allowing you to narrow down and filter your search. You can generate SBML, or Systems Biology Markup Language, to represent the data found in plant Reactome for any pathway or pathways of interest. This markup language is a common interchange format with multiple platforms, including Cytoscape. There is also a small molecule search feature found under Tools that allows you to look for small molecules as defined in the Kebi database. Search results will return to you identifiable Kebi molecules that are similar to or inclusive of your search term, and then will link to any plant Reactome data that contains that molecule. So if you look for glucose, it will give you all of the pathways in rice that have glucose. A featured pathway, currently IAA Biosynthesis 1, can be found on the plant Reactome homepage. You can link directly through the image on the homepage to the underlying pathway diagram in the pathway browser. This featured pathway will change occasionally. Taking a look at another specific pathway, this time cytosolic glycolysis, you can see in the pathway browser the details of that pathway data in the bottom panel below the pathway diagram, including reference information, the cellular compartment and compartments for that pathway, a section that gives you the ability to download that pathway data in a variety of formats, again including SBML, but many others as well, such as biopacks, and a list of participating molecules found in. If you select a reaction node within a pathway diagram, you can take a look at the reaction level data that's available. The information in the bottom panel below the pathway diagram in the pathway browser will change and show you that reaction level data, including the inputs, outputs, and catalyzers for that specific reaction. It will also allow you to link over to cross-reference sources for participants in that reaction, such as the data available at Uniprot. We have several upcoming enhancements planned for the plant Reactome, including additional metabolic and regulatory rice pathways in the next Grammy release. We want to enable more Reactome analysis tools that exist in the human Reactome that we'd like to bring over, such as gene expression overlay. And we're working to bring Arabidopsis into the plant Reactome as a reference species, so we have both monocots and dicots represented at the reference level. Finally, importantly, we want to run ensemble comparative projections to other species, including maize, so that we can extend the value of the plant Reactome to the wider plant research community. If you have any questions or comments or suggestions or would like to report issues with the plant Reactome beta website, please feel free to contact us at reactome-curator@grammy.org. To visit the plant Reactome website, again, you'll head to plantreactome.oicr.on.ca.